Hello, and welcome to this week's episode of Medisodes. We'll be taking a look at how drugs are developed. Drug development is an essential and rigorous process that is used to bring new pharmaceutical drugs to the market and clinical practice. There are several steps involved in developing a new drug. In this podcast, we'll be taking a look at four stages of drug development: drug discovery and development, preclinical research, clinical research, and licensing. First, let us look at drug discovery and development. Discovery and development of drugs is the first stage of drug development. Typically, researchers discover a new drug through a new insight into a disease process. This allows researchers to design a product to stop or reverse the effects of the disease. Technology and computer modeling has allowed tests to be carried out on multiple molecular compounds to find possible beneficial effects against a disease or multiple diseases. Another way of discovering new drugs is the development of new technologies, such as those that provide new ways to target medical products to specific sites within the body or to manipulate genetic material. When existing drugs have unexpected effects, they can be used to cure other diseases after being modified. At this discovery stage, thousands of compounds could be potential candidates for development as a medical treatment or pharmacological drug. Early testing helps to identify a small number of compounds that could be studied in further stages. Once potential drugs have been identified, the next stage is development. Information will be gathered on how the drug is absorbed, distributed, metabolized, and excreted, as well as its potential benefits and mechanisms of action. The best dosage and optimal method to give the drug, such as by mouth or injection, is also considered, in addition to any side effects or adverse events that can be referred to as toxicity. How the drug affects different groups of people, such as by gender, age, race, or ethnicity, is also vital for the drug's safety and efficacy. Other things that are considered include how the drug interacts with other drugs and treatments and its effectiveness as compared with other similar drugs. So this was just a brief insight into the FDA's explanation of discovery and development when creating new pharmacological drugs. Now on to Anupam, who will be discussing preclinical research.
So in the process of drug development, the preclinical stage, also known as preclinical studies or non-clinical studies, is the stage of research that begins before the clinical trials or the testing in humans, and during which important feasibility, iterative testing, and drug safety data is collected, typically in laboratory animals or other sources of testing. The main goals of preclinical studies are to determine a starting safe dose for first-in-human studies and assess the potential toxicity of the product, which typically includes new medical devices, prescription drugs, and diagnostics. So before testing the drug in people, researchers must find out whether it has the potential to cause serious harm, also known as the drug's toxicity. The two types of preclinical research are in vitro and in vivo. Types of in vitro testing include screening tests, which are simple and rapidly performed initial tests to determine the presence or absence of a particular pharmacodynamic activity in the new drug. For example, when testing a new pain-relieving drug, this test will allow them to determine whether it has that analgesic activity. Next, they'll move on to tests on isolated organs or bacterial cultures. These could either be naturally sourced or 3D printed. For more information about 3D printed organs, check out our episode on it on the Medisodes channel. These preliminary tests determine specific activity in the new drug, such as antihistaminic, antibacterial, antisecretory, and vasodilation. Healthy organs isolated from dead animals, or the aforementioned 3D printed organs increasingly nowadays, or bacterial cultures specially grown for this test, are used for this testing. Next, we move on to in vivo testing, where they test on animal models. When I say models, however, these refer to live lab test subjects, not fake versions of the animals. Animals such as rats, pigs, mice, hamsters, and rabbits are all used to determine the actual effect of the drug in a live organism, when it depends on which one should be used. After successful results in initial stages, higher animals like cats, dogs, and monkeys, which have systems more similar to humans, are used for preclinical trials. The choice of species is based on which will give the best correlation to human trials. Differences in the gut, enzyme activity, circulatory systems, and other considerations may make certain models more appropriate or less appropriate, depending on the drug. Also, depending on the drug's functional groups, it may be metabolized in similar or different ways between species, which will affect both the efficacy and toxicology. Increasingly, it is also possible to perform in silico profiling using computer models of the drug and its targeted interactions in the body. In silico refers to the computer chips, which are made of silicon, and these advanced computer programs can accurately predict how the drug will interact with all the systems of the body. In addition, drug development must not only also determine the toxicity and the dosage of the drug for humans, but also its physiochemical properties, its chemical makeup, stability, and solubility. Further tests are conducted. For example, experiments are conducted to determine the mechanism of the action of the drug, whether it's an antihypertensive. For example, if the drug is an antihypertensive drug, they need to determine whether it is an alpha or beta blocker, an ACE inhibitor, or a calcium channel blocker. Later, they'll look at the systemic pharmacology of the drug and its effects on individual and major organ systems like nervous system, cardiovascular system, the respiratory system, or the renal system, as well as many others. This can give a clue about possible side effects of the drug. Finally, they'll have to look at the quantitative test of the drug, where they examine the dose-response relationship, the effects of the drug, and the comparative efficacy of the new drug with existing drugs, so they can establish a market value for the drug. Of course, there will be other tests to make sure that this is as rigorous as possible, sometimes studying the movement of the drug substance in the body, how it's excreted, and many others, including how it's manufactured and how it would be controlled if it was to be produced. All this information is gathered from this preclinical testing, as well as information, as I've just mentioned, on CMC, chemistry, manufacturing, and control. And all of this must be submitted to the regulatory authorities, these being in the US, the FDA, the Food and Drug Administration, and in Europe, the EMA, the European Medicine, and in Europe, the EMA, the European Medicine Association. And this will be submitted as an investigational new drug application, or IND application. Once this IND is approved, development can finally move on to the clinical phase.
While preclinical research answers basic questions about a drug's safety, it cannot substitute ways of how the drug will interact with the human body. Clinical research is about studies or trials that are done on people. As the researchers develop the study, they have to consider what they need to accomplish for each of the different phases and begin the investigational new drug process. When designing a clinical trial, they need to follow a specific protocol developed by the manufacturer or the researcher. They need to consider who qualifies to participate, how many people will be a part of the study, the duration of the study, whether there's a control group and ways to limit bias and increase the validity of results, how the drug will be given to patients and at what dosage, and what assessments will be conducted when and where. Clinical trials typically follow from an early small-scale phase 1 study to a late large-scale phase 3 study. During phase 1, about 20 to 100 healthy volunteers with the disease or condition are tested. This study lasts for several months, and it tests for the basic safety and dosage of the drug. You don't want to be doing a large-scale trial on a drug if it ends up being poisonous or toxic. Approximately 70% of drugs move on to the next phase. Phase 2. This contains up to several hundred people with the disease or condition. So note that they are not healthy volunteers, but they have the condition. This trial stage may last from several months to up to 2 years. And this has a purpose of testing for the efficiency and the side effects of the drug. This is to see if the drug actually works for its intended purposes. If there's a marked improvement in people with the condition that it's supposed to treat. Approximately a third of drugs move on to the next phase. So phase 3 contains from about 300 to 3,000 volunteers who have the condition. And this study lasts from 1 to 4 years. And it has a purpose of monitoring the efficacy and monitoring of adverse reactions. Having a larger trial group is important because different people's bodies react in different ways. And it's important to see the effect on the drug on all people so that doctors can know if there are some people to whom the drug should not be prescribed. Approximately 25 to 30% of drugs move on to the next phase. And phase 4 is the final stage, which contains several thousand volunteers with the disease. And this is to test for the safety and efficacy of the drug. And if this passes, it can move on to the next stages. The investigational new drug process is an application to the national authority before beginning clinical research. In the application, they must include all the data from the preclinical stages. Drug developers can also ask for assistance from the national authorities during the clinical trial. For example, after phase 2, they can ask the authorities for guidance on the large-scale phase 3 studies, because these are often too large for manufacturing companies to manage on their own, especially in the case of publicizing that the trial exists and supplying payments for participating in the trial. So the clinical stages are clear-cut and simple, but they are absolutely crucial to understanding the safety of a drug and to ensure that doctors know exactly how the drug will work and exactly to whom the drug should not be prescribed. And now, if the drug does pass all of these stages, it can go to the licensing stage, for which I'll hand over to Shrey.
So the licensing stage is the fourth and final stage of a drug's development. It's where the drug company will submit an application to the regulator. In the UK, that's the Medicines and Healthcare Products Regulatory Agency, the MHRA. And in Europe, it's the European Medicines Agency, the EMA, which also covers Northern Ireland due to the terms of the Brexit deal. And in the US, it's the FDA. So these regulators will review data from all the stages of the drug's development and decide whether it's safe and effective. And if they find that it is, they will approve it. After this has happened, the drug needs to be approved for use on the NHS. And it undergoes a health technology assessment, or a HTA, which looks at its clinical effectiveness as well as its cost effectiveness, as this is taxpayers' money that is being used to fund this drug. And due to the way the NHS is run in the UK, in England, these HTAs are carried out by the National Institute for Health and Care Excellence, more broadly known as NICE. And NICE was established in 1999 by the Labour government to stop the postcode lottery that was going on at the time, where people literally across the street from each other were getting different treatments based on what their GP could afford to provide. And the NICE looks at technological appraisals of new medical products, clinical guidelines of how diseases should be treated, and also looking at intervention procedures. However, in Wales, this is the HTAs are carried out by the All Wales Medicine Strategy Group, and in Scotland, it's carried out by the Scottish Medicines Consortium. And as I mentioned earlier, in Northern Ireland, the EMA, the European Medicines Agency, regulates drugs in Northern Ireland. And this has actually caused a lot of controversy. For example, in England, Perjeta, which is a drug for breast cancer, Cornetic, which is another drug for cancer, Cententrip, Stellaris, Atalurin, Gazifaro, and Parsabiv are all available in England, but not in Scotland. And in Scotland, Avastin, Faslodex, Strivaga, which are all cancer medications, and Spinoza, which is a muscle disease medicine, are available in Scotland, but not England. And everyone, and many people have complained that drugs available should not matter where you live in the UK. It should only matter based on your clinical needs. And also, people in Wales frequently have been reported to come to England for treatment because the drugs aren't approved in Wales. And this has caused much controversy. The drug giant Pfizer has estimated that the licensing stage costs the company £15 million and can take up to 1.5 years. But there are exceptions. During the coronavirus pandemic, coronavirus vaccines have been fast-tracked and given emergency licenses due to the vast importance of getting vaccines to people as soon as possible. If you want to learn more about vaccines, please listen to Medisodes episode 1. Although once the drug is licensed, it can be used and bought. The drug development process does not stop there. Post-marketing, the MHRA has a yellow card system where people can report side effects and any faulty devices that they have. And this is extremely important in making sure that drugs are safe and that the drugs being provided aren't causing any adverse effects. An example of where this has gone wrong was the case of thalidomide, which was meant for morning sickness. The drug companies didn't realize was thalidomide caused birth defects in the babies of the pregnant people, and this caused many babies to be born with birth defects. And it was only after work by the FDA that they realized this was happening and thalidomide got banned. So now, even after a drug is being released, regulators keep a close eye on whether the drug is safe and regularly review to make sure that the drugs are still both cost-effective and clinically effective. Now back to Surya.
So this was a brief insight into how the drug development process works. Now, because of COVID and the need for vaccines, the drug development cycle has been accelerated. And do you guys think that this is an ethical approach to vaccine development?
So I think we have to make a compromise, because if we had done the full drug development cycle for the vaccines, then many more people would have died from just the spread of the disease without any treatment options. So we have to find a compromise between the increased risk of the vaccines not being safe and the increased number of people who would die. And of course, with the COVID vaccines, because of the need for it, the rate at which work was done was greatly accelerated. There was no delays in paperwork, and other work was postponed. So even at this accelerated work rate, the drug development process was not shortened that much. So you can say that the vaccine is safe and that the risks definitely do not outweigh the benefits of it.
Yes, I think Adrian make a good point there about the misconceptions a lot of people have. The COVID vaccine development process, although it was very quick, it wasn't because they'd removed certain safety steps or anything. However, it's because due to the global need for it, they were finally able to get the sort of funding that many drug development programs simply lack. They were able to get the volunteers that many drug development programs struggle to get for their programs. And I think that's why it was so much faster, rather than them cutting corners, as many people believe.
And further to this, there's still been a regular assessment of whether these vaccines are safe. For example, in the UK, there were reports of blood clots caused by the AstraZeneca jab, and this led to the AstraZeneca jab being only for people over 30. And now, because of the step taken by the Joint Council for Vaccination, the JCVI, it's meant that there are now very few cases of blood clots anymore. And this has shown how drugs and vaccines can still be monitored and regulated even after they've been approved.
So this was a short overview of the drug development process. The process is carried out to make sure that any drugs given to patients are safe and effective. For more information on drug development, visit the FDA's website. So thank you for listening to this week's episode of Medisodes. Remember to like, share, and subscribe to Medisodes for more weekly episodes.